Good morning, everyone. Welcome to today's Peter Mac Cancer Center Genomics DSP virtual launch. I'm today's host, Chen Qiao. I'm the Associate Marketing Director from NanoString. First of all, thank you for joining us at this early morning for the event. And today we have a very exciting lineup of speakers from Peter Mac Cancer Centers, Royal Melbourne Hospitals, and also WeHide. And we also had the pleasure to invite the speakers from Queensland University of Technology and also NanoString to join this lineup of speakers. Today's event is jointly organized by NanoString, BioStrategy, Peter Mac Cancer Centers, and also the Royal Melbourne Hospitals. Before we start today's event, here are some housekeeping for the attendees. If you have any questions during the sessions, please type your questions in the Q&A, which is at the bottom of your screen, where you can see in the panels. And also, if you have more questions that you would like to ask our speakers, please stay towards the end, where we have some very exciting assignments, and you can also continue to interact with our speakers. And today's part of the content for today's event will be available on demand, and so you can view some of the contents after the event. And now, without further delays, I would like to start today's event and introduce the host from Peter Mac Cancer Centers, Dr. Paul McMillan. So Dr. Paul McMillan is the head at the Center for Advanced Histology and Microscopy at Peter Mac Cancer Center. And the centers provides more than 200 researchers for the access, training, and support on advanced technology in the areas of histology, particularly on electron microscopy, optical microscopy, and also image analysis. The center provides project support in a number of spatial omics methods, including the laser capture microscopy, open multiplex immunohistochemistry, and now the NanoString digital spatial profilers. I would like to pass to Paul for today's introduction of the event. Paul, it's yours now.
Excellent. I will start to share the screen. Thank you, QQ, for the introduction. Everyone hear me okay?
All good.
All good? Excellent. Yeah, so thank you very much for the introduction, and welcome to the launch of our NanoString GeoMx DSP instrument. Before we start, I would like to acknowledge the traditional owners on the land on which we work, which are the Kulin Nation, and pay my respects to their elders, past, present, and emerging. Okay, so the reason we're here today is to launch our new NanoString GeoMx DSP system. Before we start this, I'd like to thank the contribution of the Peter MacCallum Cancer Centre and also the Royal Melbourne Hospital in acquiring the instrument. This is now available to all researchers through 2 cores within the Peter MacCallum Cancer Centre. My core, as QQ just mentioned, is the Centre for Advanced Histology and Microscopy, and also we are working very closely with the Molecular Genomics core here at Peter Mac as well. So while I've got a captive audience, I'm just going to go through a couple of slides on each of the cores just to give you a bit of background on who we are and what we do. So the Center for Advanced Histology and Microscopy consists of 4 complementary platforms. So the histology platform, which specializes in the preparation of tissue samples for imaging via microscopy, and can run either full fee-for-service or training researchers to perform their own histology. We also have a well-equipped optical microscopy platform, which has everything from slide scanners, confocal microscopes, all the way up to super resolution and multiphoton microscopy. We have an electron microscopy platform, which is again fully equipped with sample preparation and imaging for both scanning electron microscopy and also transmission electron microscopy. And finally, our analysis platform, allows researchers to access software and high specification computers for data analysis associated with all of the microscopy within the platform, and that includes 3D, time-lapse, and now spatial biology. I'll get my thing to move on. The molecular genomics core here at Peter Mac offers both library preparation and next-generation sequencing, including bulk DNA and RNA sequencing, covering most traditional genomics methods such as whole genome sequencing, ChIP-seq, exome capture, CRISPR knockout screening, and methylation pattern analysis. They also provide single-cell sequencing and are continually developing their expertise in a broad range of experimental approaches. And then finally, spatial omics, in addition to the DSP system, which is going to be discussed in this event, they also offer access to the 10x Genomics platform as well. And the molecular genomics core also integrates closely with the bioinformatics core here at Peter Mac to assist their users in quality control and analysis of their results. So as we've already heard, we've got some really exciting talks coming up once I've stopped wittering on. So we'll move on and discuss the speakers. So our first speaker up is Dr. Joe Beecham, who's the Chief Scientific Officer for NanoString Technologies, and he'll give a bit of an overview of the GeoMx DSP system and also NanoString's new spatial molecular imaging system. We'll then go on to a talk from Dr. Rachel Coldedge, who's a senior scientist at the Royal Melbourne Hospital, and she will talk about her application of DSP in analyzing the bone marrow immune microenvironment. We then have Dr. Arutha Kulasinghe from QUT, who's going to talk about a couple of projects that he's been running using the DSP, one around COVID and one around immunotherapy. And then our final presentation will be from Associate Professor Melissa Davis at the Walter and Eliza Hall Institute, and she's going to talk about her work developing bioinformatic methods to analyze DSP spatial transcriptomics in the COVID-infected lung, which is work that she's been performing with Arutha. At the end, we will have time for questions, and as QQ mentioned, we'll also release more details on the grants that are available to get you guys using the instrument. And with that, I'll pass back to QQ, and you can introduce Joe for his talk.
Thank you, Paul. Thank you very much for the introduction of the core facilities. And it's really exciting to see all the spatial technology are available to the researchers now. And as Paul mentioned, please stay towards the end of the event because we will have a research grant available through the Peter Mac Cancer Centers for GeoMx DSP access. And to learn more about the GeoMx DSP technology and how to utilize it, so please listen to our talks. So now I would like to introduce our first speakers for the day, Dr. Joseph Beecham from NanoString Technologies. Dr. Beecham is the senior VP for our R&D and also the Chief Scientific Officer. Most important, he is the inventor of the GeoMx DSP technologies, which has more than 70 peer-reviewed articles. And also in recent months, we have seen a lot of publication coming through Nature Medicines on COVID and also COVID research and as well as pancreatic cancer research. So without further delay, I would like to pass to Joe for his presentation to share with the excitement of spatial biology today. Thank you. Hi, Joe. You are mute right now. I will unmute you.
There we go. Sorry about that.
There we go. I can hear you very well.
Thanks, QQ. And it's great to be talking with all of my Australian counterparts. I actually went back and looked at some of my photos from last time I was in Melbourne. And there's a little one there where my partner in crime, Brad Brastham, and Paul Rasmussen were walking into the Peter Mac. This was about 2 years ago now, and nobody's been traveling for about the last year and a half. I also gave a talk over at Royal Melbourne when I was there, and I think I was waiting for the talk or whatever, and wandered into this archive room at the Royal Melbourne, and it was a really a fascinating place. So perhaps some of the people in the audience maybe have wandered into the archive room of Royal Melbourne. But anyway, it's great to be talking with all of you today. I wish I could be there. It's like it's Sunday night here in the Pacific Northwest, so I'm giving you my talk from my home here. So, you know, it's an exciting time to be involved in spatial biology. You know, a lot of us been working in this business for a lot of years. I remember building next generation sequencers when they were just coming out. That was Nature Method of the Year back in 2009. Then we had a great batch of technology was invented in 2012. And this was all related to single cell RNA-seq. And that really allowed us to get to the place where instead of just doing bulk-based sequencing, which you see over on the left, we could actually take it apart and actually get sort of what I call a parts list of the tissues we're examining. But then back in, you know, the regrettable aspect about single cell RNA-seq, you get a great parts list, but you don't know how all the parts fit together to make a whole function. And that's really when I started working really hard on how do you take high-plex molecular multi-omic technology and make it spatially resolvable. And little did I know when I scribbled this idea for GeoMx DSP on a little memo pad from a talk I was giving at MD Anderson on January the 6th, 2016, is actually where the heart of the GeoMx idea started. And then now to be sitting here in 2021 and seeing all this great work being done now is really satisfying. But I mean, this era of spatial biology has just begun. And I think, you know, I always find this slide got good for just sort of why spatial biology is so important and why we need these high-plex technologies there. You know, I think the key reason is how to highlight it in here. Here we have two different cancer samples. This colorectal cancer here. And we worked for years with a company called Merck on a drug called Keytruda, which was developed, that little thing, that little ring you see up there in the right-hand side called TIS. That was a bulk-based tumor inflammation signature that we developed to predict the response to Keytruda. And if you have a high number, you got a hot tumor and you're expected to respond. Now you can see both these tumors record high on the tumor inflammation score. But when you look at the spatial patterns of the cancer, they're very, very different. The one on the left is what I call a hot but excluded tumor. If you count up the immune cells, it's about 50% immune cells, 50% tumor cells. But in this particular case, the immune cells in magenta are excluded from the tumor itself. Now you look at the tissue on the right, about the same ratio of immune cells to tumor cells, but in that particular case, the immune system has been able to infiltrate the tumor itself and will be much more likely to respond to checkpoint inhibitors. Although the bulk answer, state-of-the-art answer, best you could do, actually characterize both of these tumor types as being similar. In reality, they're very, very different and would be predicted to respond to immunotherapy differently. And that was the whole reason why I realized we needed to have high-plex multi-omic technology, but it had to be spatially resolved. The concept that I scribbled down on a memo pad in Houston, Texas, back in 2016 is essentially here. The key concept was, instead of generating assays where you put the assay in the tissue, we essentially turned the whole thing around. So instead of putting the assay in the tissue, all you need is a spatially resolvable sequencing index. And that's what you see here, whether you're on the back of an antibody in the upper left, or on the back of a FISH probe, in situ hybridization probe. There's a small little, denoted in red, 60 base pair single-stranded sequencing barcode. And inside that sequencing barcode is 12 base pairs devoted to just tell me what particular antibody I'm on or what particular FISH probe I'm on. On other parts of that sequencing barcode is a couple of adapters the dual index adapters from Illumina, a universal molecular index to allow you to do unequivocal quantitation of that, and then the special trick associated with the digital spatial profiler, those barcodes are connected to the affinity reagent through photocleavable linkers. And that's the key aspect. You know, what do microscopes do for a living is they shine light in very small spaces. So by putting the sequencing barcodes on a photocleavable linker allowed us to develop a general purpose system. And you can see that over on the right, the GeoMx Digital Spatial Profiler. We put 2 semiconductor chips in the back of the microscope that would allow me to put the UV pattern that would release those photocleavable barcodes. That UV pattern, those mirrors will adapt to the exact geometry of the tissue you're examining. So every tissue is different. If you look at it in a microscope, those mirrors will respond to the geometry associated with every tissue and allow you to get pure, say, whole transcriptomes of the tumor and tumor microenvironment that are uniquely spatially resolved with respect to the geometry of the tissue itself. I'm going to just take you very quickly through the overall cycle on how the GeoMx works. So you can see the DSP that's now down there at the Peter Mac over on your left-hand side of your screen. This technology was built from day one to do biobanked FFP samples. So all of the chemistry we developed came out on FFP. That's where we developed it. That's where it has the highest sensitivity. So there you start out with a normal FFP slide, a biobanked FFP slide. Those barcoding in situ reagents, whether they're antibodies or in situ hybridization reagents, are invisible, so you don't see anything. So you dope in 4 colors of fluorescent signal at the same time you put all the high-plex antibodies or the high-plex in situ hybridization probes on your typically a 5-micron thick FFP sample. And step 2, you use the colors associated with the 4 colors of fluorescence to get the overall tissue geometry. And that allows you to actually look at the geometry and select those areas that you really want to examine for high-plex profiling. And I'll show you a lot of the selection capabilities during this talk. You find yourself to a unique region, step 3. You emit the UV light. Takes about a half a second to photocleave all the photocleavable barcodes off the antibodies or the fish probes. They are collected off the tissue surface with a small microcapillary, then deposited onto an on-instrument microtiter plate. This entire process of actually photocleavage, putting them into the microtiter plate and going to the new location takes about 45 seconds. So you can wander through the tissue, generating what I call high-plex digital replicas of the affinity reagents and how they're distributed in space. Realize this is a non-contact technology. The sample is preserved. When you're done with it, you can pull your sample back out of the instrument, store it wet. You can actually go back and do it again if you wish, or use it for some other type of analysis you want to do. So it's non-destructive. When you're done with collecting your areas of illumination, all regions of interest, you got these digital replicas. They come out of the instrument. And on proteins up to about 100 plex, you can read them out directly on a normal NanoString nCounter instrument, single molecule optical barcoding instrument. When you go to high plex or ultra high plex, and currently I'll show you data up to 22,000 RNAs in space, you read that out on an Illumina sequencer. The instrument keeps all the high resolution images, sequencing data, and nCounter data, keeps track of all them, and then brings them all back. So you always end up with high resolution images coupled to your high plex data. This gives you an idea of what the current content is on the GeoMx Digital Spatial Profiler at this time. We have over 300 antibodies that have all been optically barcoded with these photocleavable barcodes, emphasizing particularly immuno-oncology and neuroscience detection. That's the protein-specific targets, and I'll show you quite a bit of the results there. And then over on the right-hand side, you see our spatially resolved RNA. Back in 2019, actually it was 2020, we came out with what's called the Cancer Transcriptome Atlas, which is a little over 1,800 genes in space, transcripts in space. And these were all developed from the TCGA database. All the high variant transcripts related to key cancer biology and tumor biology are brought together in the Cancer Transcriptome Atlas. Now, just this year, in March of this year at the AGBT meeting, we launched commercially both the Human Whole Transcriptome Atlas, which is 18,500 over 18,500 genes in space. And then we also, coming out this quarter, is the Whole Transcriptome Atlas for mouse, which is over 22,000 genes in space. And I'll show you what the data looks like. Now, very important, I remember when I was doing that work and inventing that with a professor at the MD Anderson Cancer Center at that time. His name was Gordon Mills. He said, Beecham, if you're going to make a high-plex technology spatially resolved, it's got to work on biobanked FFP samples. So that's what we did. All the chemistry was designed. Works on FFP tissue blocks, and we've looked at blocks as many as 35 years old. Still get good data out of them. Standard slide mounting, could be a tissue microarray, could be a core needle biopsy. We've done fine needle aspirates. It could be fresh frozen tissue, of course, if you have that. But there's no need. Totally designed with biobanked FFP samples in mind. And one of the key aspects is that you do your high-plex profiling in a shape that's linked to the geometry of the tissue you want to investigate. That's really important because each tissue is different, and it really allows you to perform analyses that are just simply impossible to do in any other manner. And then, so those semiconductor mirrors that are in the back of the GeoMx will auto-configure in all these kinds of patterns. And we actually give you control. You can even make your own patterns up if you wish. But we hardwired some in there. This one called segmentation I find very advantageous for doing tumor microenvironment. The instrument will know how to actually illuminate only the tumor-specific cells in an automated manner. The mirrors will adjust, then select only the tumor microenvironment, and you end up getting pure whole transcriptomes on those two compartments, say. It'll also find rare cells where you actually want to do profiling on a bunch of rare cells in a particular field of view. You can do that. Then we got this area called contouring, which is very interesting in the area of immuno-oncology. I'll show you in detail because you can actually set up the mirrors to follow the geometry of exactly where two different tissue types or cell types come together, which is where a lot of the biology associated with immuno-oncology it comes about. Or you can just sit there and draw with a mouse and do your own sketches to do your area of illumination. This gives you an idea how the mirrors conform to the shape of the tissue. This is a colorectal sample we're looking at here. And we're we're doing whole transcriptomes, and I want pure whole transcriptomes on the tumor, and I want pure whole transcriptomes on the tumor microenvironment. So we're sitting here looking at a pan-cytokeratin antibody stain it's shown in yellow, and it will light up only the tumor-specific cells. The mirrors will adapt to exactly that geometry. You can see that in the upper right here. That forms what you call a mask. Those mirrors will now reflect the UV light in a pattern that's with single cell resolution, just maps the tumor-specific cells. You get all the transcripts coming off of that. You put it in one well of the microtiter plate. The mirrors will now invert and adjust. You can see that mask on the bottom, and it will re-illuminate. And this time, all the UV light's going to be directed directly to the magenta component, which is the CD45 positive region. So that will be all the immune cells there. You flash the UV light, then you got pure whole transcriptomes coming out of the tumor microenvironment. 2 45-second operations. And this is what it looks like when you do that. I'm going to show you a bunch of protein results and then switch to the high-plex RNA results. This is a particular example. This is what non-small cell lung cancer looks like on the GeoMx DSP. This is work done at the pathology group at Yale by David Rim. And David's interested in looking at the tumor-specific pancytokeratin, the immune compartment CD45, the macrophage compartment CD68. You can see also he's interested in non-immune containing stromal regions. So he compartmentalizes all aspects of that particular non-small cell lung cancer tissue slice, illuminates in that pattern of 4. This recently came out in Clinical Cancer Research, so I'm not going to go into huge about it, details. What David was able to find was 12 protein biomarkers that were associated with predicting response to checkpoint inhibitor called Keytruda in this particular case. You can see the ROC curves over there on the right, using the spatially resolved patterns and the protein components associated with those particular spatial compartments to predict response to checkpoint inhibitors. This has been done a number of times now. This is another paper that just recently came out from the Huntsman Cancer Center related to triple-negative breast cancer. Similar thing, using our high-plex protein technology to try to predict responses and prognostics of triple-negative breast cancers and how they respond to therapy. Now, we've had a number of successes along these lines using high-plex immuno-oncology targeted protein panels, typically about 50-plex out to about 100-plex. And actually, I went back to our original first 2 papers that were ever accomplished using this technology, showed up back-to-back in Nature Medicine. A co-author on this paper was Jim Allison, who won the Nobel Prize for his discovery of CTLA-4 and broke open a lot of the whole checkpoint inhibitor drugs that are now widely used in here. He was actually a co-author on the very first DSP paper. In fact, when we were doing this, it was high-plex protein we were doing, and the investigators were mainly interested in T-cell biology. But we had a panel to have both the B-cell component and the T-cell component in there at the same time. They told me when they were starting that study, I'm really only interested in the T-cell component. But it turned out some of the most informative predictive hits that they found actually was from the B-cell component that was in our panel. And it was associated to these unique structures in cancer called tertiary lymphoid structures. This was really one of the early papers showing that tertiary lymphoid structures are predictive for response for immunotherapy. They went back after we published those are published back-to-back in Nature Medicine. They literally turned an entire whole study in less than 9 months on the GeoMx DSP and actually followed that up with a paper in Nature. There was actually back-to-back papers in that Nature one also, showing the role of tertiary lymphoid structures. And all these came out of the high-plex protein capabilities of the GeoMx being performed in this particular manner. It's a number of papers out now, and I just highlighted some of them. Probably what I'll highlight, if you really want to look at the detailed validation work of all the technologies for both the protein and the RNA level, you can read our Nature Biotech paper from last year. We'll go in if you want to look at all the supplementals, all the detailed things you have to do for verification and validation of that technology. And the papers literally come out every week. I sort of keep a track on them. I think one of the key things are the work that's being done, predominantly about 80 or 90% of our papers at this stage is in the area of cancer. Cancer predicting response, mechanism of action of biomarkers, developing new drugs, new therapeutics, new diagnostics are in the area of immuno-oncology. And you can see these papers are passing, if you ever had to have a paper in Nature, you can see there's 4 of them now in Nature showing DSP data in there. You know what kind of reviews they have to look at every particular way you collect the data, so on and so forth. And all those top tier papers obviously are really very difficult to get peer reviewed work in, and the GeoMx has been doing great at this level of peer review and this kind of level of information. So since I'm talking to this group, I thought I'd throw in a couple of things I don't normally do in terms of, is there a path of GeoMx towards the clinic? I actually thought it'd probably be another 5 years before I was talking about this, but it's actually starting to happen now. a couple of areas of development shown up, and let me show you a little bit of how that's worked. You probably know about NanoString. You know, we have a long history of taking even our bulk technology, nCounter technology, make translational discoveries, typically in cancer, and then actually translate those discoveries all the way into clinical grade assays. We've done that a number of times. You can see here, we have a whole FDA cleared breast cancer diagnostic we developed that way called Prosigna. And we've done it together with Celgene for a drug called Revlimid and DLBCL. We developed with Merck. I showed you the tumor inflammation signature with Keytruda. We also had another one for a drug called enzalutamide that we developed with Medivation and Astellas. All these were robust assays that made it all the way into phase 3 clinical trial work. Now, it's interesting to see how GeoMx progressing. Actually, I was a little scared when I first knew this study was going to be done because it's a high bar. David Rim in pathology took our high-plex, it's like a 50-plex protein assay that we do, and it has one of the targets in there is PD-L1. So we have a bar-coded PD-L1 antibody in there. And he ran it head-to-head against whatever, 3 FDA-cleared 1-plex assays for PD-L1, and then 1 laboratory developed test for that, and ran it head-to-head. All he's doing is taking 1 out of the 50 protein barcodes and just plots that with respect to the 1-plex DAB assays that the FDA cleared. And you can see tremendously high correlation on the barcoding technology of GeoMx compared to those 1-plex high resolution FDA cleared assays. So I'm pretty happy. He was very happy about that kind of performance, and so was I. And then it continues in this direction. The Knight Diagnostic Laboratories have now validated 82 of our antibodies. They are actually in to making decisions at the tumor board for a specific set of clinical trials work that they have at OHSU. We're also partnering with OHSU and also with the Mayo Clinic to develop some unique spatially resolved assays for breast cancer development. That's a work in progress. We had a bunch of posters at the AACR that's just happened. This is happening, I guess. This gives you an idea what this is: analytical validation of some of our protein content at the level of a CLIA lab, and I just sort of put that on there. We also have phosphorylated components as well as non-phosphorylated components, and that they're actually in breast cancer. They're using some of the phosphorylation status to answer some very unique questions in terms of response to therapy downstream of EGF receptor signaling. Very interesting area. Okay, let me show you what high-plex RNA profiling looks like. Literally in 2019, when I remember doing this talk, and our very first panel was 84 genes at that time, and then we had another 16 controls or something. But it was the largest RNA panel commercially available by about a factor of 5 or 10 when we launched that in 2019. We didn't stop there though. The next year we did 1,800 genes in space. And then this year, early this year, we totally launched the whole transcriptome, mouse transcriptome. So we're up to 22,000 genes. So we've gone from 84, which is best in commercial class 2 years ago, to 22,000 just in the last 2 years. It's nice to be able to develop it, and I'm going to describe to you pure information content of whole transcriptome profiling of these cancer tissues. I think is really amazing. Now, here's a couple of technical details about the whole transcriptome assay. It's not a poly A pull down. So poly A pull downs, all your sequencing dollars go into these high population transcripts that actually suck up about 35%, 40% of your entire sequencing bill are these mitochondrial transcripts, and the top 10 like GAPDH and GUSP and all these other. With non-poly A pull down, I don't have to waste all that time and money on those. I can go right deep into the key transcriptome of biomarkers that are really important there. they view all that from sequencing mitochondrial genes. You can add them back in if you want customized. I just didn't want you to spend all that sequencing money on them. Works on fresh, frozen, FFP, biobank samples. You can customize, you can drop your own little targets in there if you wish. I'm going to show you the kind of information content that comes out of a whole transcriptome assay. Now, one of the things when you go to this high plex, it's important for us to not sacrifice anything with respect to the reproducibility and accuracy of the technology. And this is what you see here. This is multi-user, multi-instrument validation of over 18,500, the whole human transcriptome, repeated multiple times on serial sections, different users, different instruments. The reproducibility here is as good as our clinical grade bulk assays that actually made it all the way into phase 3 clinical trials. We're not giving up anything with respect to the robustness and accuracy by going to this higher plex. Huge dynamic range here, high sensitivity to both low, medium, and high expressors. And then you have this unique ability to get compartmentalized, unique geometries associated with the tissue sample itself, whole transcriptome out. You're not mixing up tumor and immune cells with 18,500 genes. You get these unique transcriptomes of the immune compartment and the tumor cells only compartment. And that's real data there. You can see the shapes of those transcripts in terms of their population and weighting are very, very different. And it's really important to be able to resolve them in that manner. Now, this is what it looks like when you're in a real excisional biopsy now. And I wanted to be able to answer for people how many genes you're going to see in a typical 5-micron biobanked FFP sample. I'm doing about 100 whole transcriptomes on this particular colorectal sample here. And again, these are whole transcriptomes that are compartmentalized into the immune compartment and the tumor-only compartment. Each one of those is a 300 micron circle cut into tumor and tumor microenvironment compartments. Each of those compartments are going to yield about 9,000 genes out of the whole transcriptome. And that 9,000 genes got a ton of information associated with them. Let me show you how they change in space. These are whole transcriptomes. When we wanted to organize them in a way that you could actually look at them with your eyes. Some people think when you get high plex, it's going to be hard to analyze. I think it's exactly the opposite of that. By looking at entire pathways, I think it's a lot easier to analyze when you have this kind of information content. So we take the whole transcriptome, put it in a Surcos plot, and then organize that Surcos plot in terms of the pathways, like the dial around a clock. You can see cytokine, chemokine, extracellular matrix signaling up here between 12 and 1, TGF beta, TLR, TNF signaling down here around 2 o'clock, and you can just organize it this way. I'll put the tumor whole transcriptome and the tumor microenvironment whole transcriptome, and we just watch as it changes as I move across the tumor surface. You can see some of that over on the right-hand side. And you can see a particular area like cytokine signaling goes up and down as I just move 1/10 of a centimeter, a millimeter around on this roughly 1 centimeter by 2 centimeter excisional biopsy that I'm looking at here. And this information content is really dense and very useful. I'm going to blow up just one of these whole transcriptomes. It's the cytokine chemokine. And you can see that one region of interest over here is hot in terms of the overall cytokine chemokine response. I move a little bit over there to the left, about 1 millimeter or so, and you can see that the tumor over in there, the tumor microenvironment is cold there. So you got the difference between a hot and a cold. It's literally a millimeter away from each other in the same tissue excisional biopsy. And you can watch the whole pathway set change as you move around the tumor. I'm going to highlight a different kind of pathway over here. This is work we did with the Broad and Mass General Hospital. This is also colorectal cancer. I'm in an area that's a low-grade tumor. Here's the Surcos plot. And pay attention to this T-cell exhaustion area, this IDO1 cluster. Down there, there really isn't a lot of T-cell exhaustion right there. If I move down to this area that's the high-grade part of the tumor, you can see the whole IDO cluster increase associated with T-cell exhaustion, and I follow that into a lymph node metastasis, and it carries that T cell exhaustion IDO1 cluster with it as it travels. Again, just highlighting the detailed information that you have in these studies at the whole transcriptome level. Now I want to highlight this one because this has to do with the fact that the biology of the tumor microenvironment, here the tumor cells are in green, the immune compartment's in magenta, and I'm looking at that boundary where the tumor cells and the immune compartment come together. I'm going to orient these mirrors to make little 50 micron wide expressway lanes, if you will. And the mirrors, one at a time, I'll collect that one expressway lane. I'll be doing whole transcriptome on that, or cancer transcriptome atlas. And then I'm going to turn all the mirrors, get the whole transcriptome there, move over here into the tumor and get that whole transcriptome. And do that as I move through the tissue geometry. And I look at the pathway information. Over here, look at the MET. MET's something linked uniquely to the tumor-only cells. And you can see they're high. The minute I go off the tumor mirrors, jumping into the immune compartment, they go down to near zero. Then look at over here, something like MHC class 2 antigen presentation. Look at what happens there. When the immune compartment hits the tumor compartment, you get this whole burst of MHC class 2 antigen presentation that follows the exact geometry of where those two compartments meet. And I can follow that biology exactly in space. Of course, you got hundreds of other pathways. You can make simultaneous plots, overlay them on your class 2 antigen presentation, yielding an amazing amount of information that you can get from these kinds of samples. Now we have to speed it up here at the end here. One of the things when COVID hit, actually, the GSPs, I run 4 or 5 of these up in my labs here in Seattle, 24/7 for samples coming from the outside. I think probably several of you probably have run samples through our technology access programs. When COVID hit, we were almost the only discovery technology that could actually take these COVID samples. They were highly fixed samples. They needed the information with a rapid turnaround time, and they needed discovery. We left the labs in Seattle open. The rapid autopsy programs from about 10 medical centers from around the world contacted us and started sending us samples. This work is now showing up into peer-reviewed literature, so I can start talking about it. Actually, one of our very first papers was with Mass General Hospital, showed up in Nature Communications. Followed that up, Weill Cornell in New York City was another one. This one showed up in the journal Nature just recently. This was where we did an entire atlasing of COVID-19 progression. And just to give you a highlight, when you got high-plex RNA, you can really go in there and discover new biologies. And I think maybe Arutha is going to talk in the same series, so I'm not going to show you a bunch of this, but this is what COVID tissue looks like. You can go in and image the virus, do whole transcriptomes all around it, and determine some amazing biology associated with this. Now I'm going to end on just one thing. This isn't really a cancer-related thing, but when I saw this data, I thought it was so validating of what you can get with whole transcriptomes in tissue. I just wanted to show it to you quickly here. Just go into something like a kidney, and I'm going to actually do whole transcriptomes, spatially resolved, using the little mirrors to follow the geometry of the kidney tissue. I'm going to look at whole transcriptomes of glomerulus, proximal convoluted tubules, distal tubules, loop of Henle, and all the way out to collecting ducts, and say, how's that work? What's the information content? There's your tissues. There is the mirrors are going to configure to generate these unique substructures as I go through the kidney. And I'm not going to tell you about kidney biology, but suffice it to say that with this one experiment, we recreated about 50 years of physiology that's been done in the kidney. All the transporters we thought should be in one area of the kidney show up exactly if they're in the proximal tubule areas. You see all the sodium glucose co-transporters, all those things. And then when you move down into the distal tubules, you see the ammonium and bicarbonate show up exactly in space where the kidney physiologist told us they would be. And then what's really interesting is you go into the transcripts themselves. we've been validating the transcripts that we see against the only method that I know of that's got this kind of spatial information, the Human Protein Atlas. And here I can plot out like where in the whole transcriptome, where the gene for CALB1 shows up. It only shows up when I'm in the distal tubule part. If I go into the Human Protein Atlas, lo and behold, that's exactly where that protein is in the kidney. It's the only place it's there. And then here the transcripts associated with the whole transcriptome only show up when I'm in that spatial compartment. And you can just go through the whole thing and see all the transcripts mapping to the Human Protein Cell Atlas in a spatially resolved manner. So it gave me a lot of confidence that this whole transcriptome is really following the biology of a tumor, and then you can go into a brand new tissue and recreate pretty much all the physiology of these tissue samples you want nearly instantly with this information content. So I know I'm just a couple minutes over. I'm going to stop here. And anyway, hope this gives you a good idea of the technology that's coming to the Peter Mac and the extended community. They can use it. And yeah, good luck on your research. And I'll be sticking around for the rest of the talks. Thank you, guys.
Thank you, Joe. Thank you so much for the exciting talk. It's really amazing to see the data to publication time is so short on the GeoMx DSP. There were lots of questions coming through the Q&A from the audience. Due to the limit of time, I would like to ask you a couple of questions before we move on to the next sessions. But for all the audience, for your questions, we will address them at the end of the session and also individually. So the first question where a lot of audience are interested is the resolution of GeoMx DSP. So the question is how many cells, what's the minimum number of cells you can capture with GeoMx DSP, and what's the smallest area you can work on GeoMx DSP on the tissue type?
Question. The hardware is built with a spatial resolution of 1 square micron. An individual mirror in the DMD actually subtends 1 micron by 1 micron. So you can illuminate that small area if you wish. With proteins, sometimes you go all the way down. It's not too hard to go all the way down to single cell limit with protein-based detection. And you can look up the protein limits in some of the papers we've done. They typically are 1 to 4 cells or so. With the RNA is less sensitive than the protein-based assays. And again, it depends on what you're examining. If you just want to give you a rule of thumb, on the order of about 50 or 100 cells in a unique spatial compartment gives you pretty good signal out of the whole transcriptome assay. I've seen a couple of papers that are coming out that went across my desk where they got whole transcriptome on 4 cells in the human brain. That's going to be published soon. So you certainly can see, you get signals out of those small cell numbers. Again, it all depends on, the expression level of the markers you're interested in. So it really depends. I've seen papers cross my desk with as little as 4 cell whole transcriptomes, but typically it's on the order of 50 to 250 cells for these whole transcriptomes, typically.
Thank you, Joe. Thank you very much for the answer on the resolution. I think the second question where a lot of audience are interested in is about the tissue type. So the question is regarding, is GeoMx DSP like protein and RNA application be optimized on all tissue type? And how about some tricky tissues like TMA, organoids, and even bones?
Yeah, that's a great question. So what you're going to find is that the behavior of the whole transcriptome atlas has been designed to work on pretty much all types of FFP tissues. So I think over the course of the last 3 or 4 years that I've been running whole transcriptome, I think we've looked at about 56 different tumor types. These are real samples that are coming in from the outside into my labs here in Seattle. Yeah, we've been successful on all these different tissue types, even things like bone marrow samples. And I think maybe there'll be some bone marrow examples in some of the talks today. Very successful. Just finished a huge project with pancreas. And a lot of times pancreas is really notorious for being a difficult sample to measure in. But remember, our technology is non-destructive We never have to grind the tissue up. So we get absolutely gorgeous high-plex signals out of pancreas, even though if you actually do the bulk and some of the bulk sequencing and even the single cell work, there's so much enzymes there that degrade, but it actually doesn't cause our in situ non-destructive methods any problem whatsoever. I've looked, like I said, I've done proteins all the way back to 35-year-old protein samples. On RNA whole transcriptomes, I think I've done up to about 25 years. The question is not so much how old the FFP bank is. It's when you cut it. So for proteins, don't care. But if you got an old FFP tissue, if it's not been cut and sitting around on a slide for 20 years, if it's been cut and then you measure it within a few months of cutting it, you're going to have a high probability of success, regardless of how long that FFP tumor may have been banked.
Thank you, Joe. Thank you very much on explaining the years of the samples and also the type of samples. And thank you very much for the exciting talk again, and joining us from the United States and on your Sunday night. Now we will move to the next talk, as Joe had mentioned, where our bone marrow sample is an application. Now let me pass back to Paul for today's agenda.
Thanks, Joe, and thanks, QQ. So our next speaker, Rachel, do you want to get your slide up? Our next speaker is Rachel Colditch. She's Royal Melbourne Hospital Fight Cancer Foundation Faye Duncan Women in Research Fellow and also a senior scientist in the ACRF Translational Research Laboratories at the Royal Melbourne Hospital. And she has pioneered the application of DSP to archival bone marrow trophine samples. And we're going to hear a bit about that work today. Over to you, Rachel.
All right, so thanks, Paul. Yes, I'm going to be talking about application of DSP in the analysis of the bone marrow immune microenvironment. So as Joe very well covered, in recent years, understanding of tumor immunology and microenvironment has led to the development of immunotherapy. Things like CTLA-4 and PD-1 inhibition have really driven the treatment of solid malignancies. But analysis of the bone marrow microenvironment has been limited. And this is because most analysis of the bone marrow is restricted to the use of bone marrow aspirates, which lose their microenvironmental context, and in addition can have some sample complexities. If your patient has a significant tumour infiltration, your bone marrow aspirate really is going to be 90% tumour cells, so you can't really analyse your immune microenvironment within that. Conversely, if your patient has bone marrow failure syndrome, their bone marrow may be completely empty, and your aspirate may give you no cells, so you can't do any kind of analysis on that. And therefore, there is a need for techniques that allow us to analyse the bone marrow microenvironment in clinical samples that allows us to identify new biomarkers of treatment response, new drug targets, and potential cures. So bone marrow trophines are a core biopsy of the bone marrow. They're taken from every patient with a hematologic disease as part of their clinical care. And their processing is designed to preserve the morphology of the bone marrow. So they undergo a harsh acid decalcification step in addition to being FFPE mounted. So this destroys any DNA or RNA within the sample. So we can't do the very nice spatial genomics work that is now coming out. We're restricted to analysis of the protein. And today, immunohistological studies on these samples has been pretty limited. But there are lots of archival samples from these patients. There are 30+ years of samples in pathology departments worldwide. There are untapped resources for translational hematology, particularly in patients with fairly rare diseases. So we were the first to apply GeoMx to bone marrow trophine samples, which we published last year. Just to give you a bit of an overview of that study, we analysed just 5 patients 1 normal, 2 patients with aplastic anemia, which is a bone marrow failure disease where the T cells become activated and destroy the healthy bone marrow, leading to failure of the bone marrow. And 2 patients with myelodysplasia, which is a cancer of the myeloid lineage. These are all diagnostic bone marrow trophines collected through routine pathology, and we selected 12 regions of interest per sample, 300 microns, detected by dual CD345 positivity, so that we could focus on the immune infiltrate. And just to point out here, what we see in bone marrow diseases, even in cancers, you don't see what you would see in a solid organ cancer. You don't see a big mass with areas of cancer versus areas of immune or normal tissue. It really is all dispersed together. And these big empty spaces you see, they're the fat storage within the bone marrow. So that's completely normal to see this kind of patchy lattice work of cells. So these are representative ROIs from samples. And as you can see, this MDS2 sample was had a high level of tumour infiltration, whereas the aplastic anemias had very low levels of cellularity. And so we had a range in cellularity anywhere from 5 to 70%. So we looked at quite a wide range of cell densities within these samples. And what we saw was that DSP was very good at discriminating between the aplastic anemia and MDS samples. By heat plot, the aplastic anemia samples were segregated away from the other samples, and we saw that expression of CD14 and VISTA were differential between the MDS and the aplastics. The CD14 differential expression has been previously reported in publications, but the VISTA expression downregulation in aplastic anemia is something that hasn't been previously reported, and it is a very interesting target because VISTA is involved in autoimmunity, and it suggests that it may be a way that the T cells within patients with aplastic anemia become activated and destroy the healthy bone marrow. And this is something that we're following up right now in some of our extension studies on aplastic anemia samples, and we've got DSP data waiting for bioinformatics analysis, so hopefully we'll have some validation of this result soon. So I'll move on to a little study we've done in hairy cell leukemia. This is a very rare CD20 positive B cell malignancy. The patients have fairly rare circulating hairy B cells, but they have extensive marrow infiltration. So the marrow is the primary site of the disease, and it accounts for only 2% of all leukemias diagnosed each year. So it is quite rare. Fortunately for hairy cell leukemia patients, they have a fairly good frontline drug that is used, which is the purine analog cladribine. But for these patients, the duration of remission can be highly variable, anywhere from 1 year to 20 plus years. But for all of these patients, a cure is very rare, and 50% of patients relapse within 10 years of treatment. So in this study, we used DSP to analyse 9 patients with matched pre-post cladribine samples available and 10 healthy controls. So these are the representative ROIs here, and you can just see at diagnosis these, the green is CD45, which actually in this disease is the tumour cells. The hairy cells are 45 positive. So you can see the extensive infiltration of the tumour at diagnosis, whereas at post-treatment it's not resolved completely, but is certainly a much lower tumour burden. We sent this data to Melissa Davis and her team for bioinformatics analysis, and she was kind enough to help us out with that. And we have 3 comparisons that we looked at, post versus pre-treatment in the hairy cell leukemia patients, and pre versus the healthy control, and post versus the healthy control. And what we see is that there are lots of different proteins that are up and down regulated. but there is a very significant difference between pre and post. The most significant p-value is to the -15, and same with pre versus healthy. But post versus healthy becomes much more towards a normalization of immunity, with the p-value being nowhere near as significant. And just to pull out a few of the significant results, so just a few of the tumour markers, CD20 and CD11C, we see that at diagnosis in red, they're at high levels, which decrease with treatment in green, but they don't decrease to the level of healthy control in most patients. So most patients still exhibit ongoing tumour cells. And looking at a few of the immune lineages, we see that there's no real change in T cell lineages, and here I've just shown CD3, but multiple myeloid lineages, I've just shown 14 for an example, multiple myeloid lineages were increased with cladribine treatment, and this is backed up by studies that have been done in patients with multiple sclerosis who are also treated with cladribine, where there's an increase in various myeloid lineages with treatment. The thing we were most interested in was biomarkers of response to cladribine, and thankfully we had 4 patients who responded to cladribine and 4 patients who didn't, allowing us to do this analysis. And we see that the degree of tumour depletion, so depletion of CD20, and the upregulation of T cell markers, so CD3 and 28, is associated with durable response to cladribine. So with those 2 markers, CD20 and CD8, we can develop quite a simple model, which I've just have a bit of a figure for here. So in blue, we have our hairy cell cells, and in yellow, we have our CD8 T cells. Patient gets cladribine treatment. In the non-durable responder, we see that there's a decrease in the tumour burden, but the T cells really don't do anything. They just sit there. And at long-term follow-up, which could be 2 to 5 years after treatment, the patient will eventually relapse with the tumour outgrowing in the bone marrow. Whereas in durable responders, what we see is there's the decrease in tumour cells, but also an increase in the number of CD8 T cells. They're responding to the tumour antigens. And there's this ongoing response. They may not clear the tumour completely, but the T cells keep it under control. This is something that we're now looking to validate in a bigger study, obviously, but it allows us to ask other questions. You know, can these non-durable responders at 3 months, if we can identify them at 3 months before they relapse, can we come in with another therapy that would change them from this relapse to an ongoing response? Is there something that could help that immune system, some kind of immunotherapy perhaps, that would stimulate the immune system, allowing these patients to have an ongoing response to treatment? So to conclude this, hairy cell is associated with immune dysregulation in the bone marrow, and this generally doesn't recover with therapy. Post-CDA durable responders do show evidence of increased antigen presentation and reduced tumour bulk, and application of these biomarkers could allow early identification and increased monitoring and treatment using novel therapies in patients that are at risk of relapse post-cladribine. More widely, GeoMx has allowed us to analyse the immune microenvironment in bone marrow trephines, and it's a technique that we're increasingly incorporating into our studies across various bone marrow associated diseases. It allows us to analyse hematopoietic diseases at an unprecedented level of detail, allowing us to analyse primary archival samples in some very rare diseases where these kind of analyses would previously be unable to be achieved. It allows us to construct large sample sets with high statistical power, and importantly, all of these samples are associated with long-term outcome data for correlation. It also gives us new mechanisms of disease pathogenesis and therapeutic leads, and allows us to identify diagnostic and prognostic biomarkers. And with that, I'll thank everyone in the lab who's heard me talk about DSP for about 2 or 3 years now. But also our collaborators, Melissa Davis and Chinwe Tan at Walter and Eliza Hall, their help with the bioinformatics, and our funding sources, Bright Cancer Foundation, Royal Melbourne Hospital Foundation, who were instrumental in helping get this instrument into the Peter Mac building, and also Maddie Arts Vision, who are funding some of our ongoing research. Thank you.
Thank you, Rachel.
Thanks, Rachel. If there's any questions, feel free to type them in the chat and the Q&A. Maybe while we're waiting on those coming through, since you've used the system for a fair while now, Rachel, is there any advice you would give to people starting up afresh in terms of, what can be technically tricky? What are the key things to think of before you start your experiment?
Yeah, so I think I've used one of the more difficult samples to analyze. So I think for most people, it is fairly straightforward to use. The best bit of advice I would give people is be clear about what you're trying to analyze. You need to have some really specific groups of patients that you're looking at, pre-treatment, post, X number of days or months post-treatment, whatever's appropriate for your analysis, and also source that clinical information to make sure that you've got the correct response data, you've got the correct variables for correlation that you are going to need, because it can be an expensive exercise if you do all of your analysis and then go to find that data and find, they didn't record what they should have recorded and therefore that data is missing. So pre-planning and making sure that you have the data available is essential.
Great. I don't have any other questions coming through the chat or the Q&A, so thank you, Rachel. And I think we'll move on to our next speaker, who is Dr. Arutha Kulasinghe. Hopefully I'm saying it right, Arutha.
Yeah, that's fine.
Close enough. In 2019, Arutha was awarded a Peter Doherty NHMRC Early Career Fellowship. He is based at the School of Biomedical Sciences at the Queensland University of Technology. And as I mentioned in the first introduction, he's going to give us a couple of streams of his research that use the DSP system. Thanks, Arutha.
Thanks, Paul. Can you hear me and see my slides?
Yeah.
Perfect. Thank you. Thanks, Paul, for the invite and QQ. And it's great to see Rachel and Joe and Mel at this seminar. So I'll just talk about some of our work in the last almost 2 years, I believe, in immuno-oncology, head, neck, and non-small cell lung cancer, but also some work we've done in COVID-19, which is now sort of my lab sort of split between the two.
So while Melissa's getting her slides sorted out, I'll give her a brief introduction. So Associate Professor Melissa Davis is the co-head of the Division of Bioinformatics at the Walter and Eliza Hall Institute. And she's been optimizing bioinformatics methods for the analysis of spatial transcriptomics data using the NanoString DSP. And I think both of the previous talks have used your pipeline for analysis, Melissa.
Okay, has that come up now?
Yep, that's great. Thanks.
Perfect. All right. Well, thank you very much. So what I'm going to be talking to you about today is our work looking at the bioinformatics challenges of analyzing spatial transcriptomics data generated from this platform. And it's been a real pleasure for me to work with both Rachel's team and Arutha's team. And so the work I'm going to be talking about today has been a really fabulous collaborative project that's been led by Arutha, but has involved all of these wonderful people. So Arutha obviously spearheaded a lot of the work. Chinwe senior postdoc in my group who's been working on the analysis here. And then we have a team of people up in Queensland led by Christy, who's a virologist, and Fernando and Gabrielle, both immunologists, who've been working on the interpretation and analysis of the data with us. So this has been an absolutely brilliant collaborative project, and one that I think we were all really pleased to throw our efforts into in 2020. We also have a number of collaborators in Brazil who Fernando has brought in, and most of the samples that I'll be going through, in fact, all of them, I think, have come from that Brazilian collaboration. So I don't need to tell this audience how dreadful COVID has been, but suffice it to say, we are now at a point where we're seeing second and third waves of infection. And so regardless of the presence of vaccines now that have been developed, there still remains a very pressing concern about how we can diagnose and triage patients who are diagnosed with COVID-19. So 42% roughly of COVID patients are going to develop severe respiratory distress syndrome. And of course, there are questions around what drives this in some patients rather than others. And one of the things that we clearly see is that there is a clinical need for biomarkers to aid in patient triage and treatment. So this remains a pressing concern for us in terms of deciding how we can actually treat patients. Now, looking at spatial transcriptomics data is a really ideal way to try and get a handle on what's going on across a complex and heterogeneous tissue like lung. In a lot of the biomarker work, whole blood has been used. And so the tissue that we have, which has come from autopsy, is much less common. Now, there have been a couple of papers. At the time we put these slides together, there were 2, but there's probably a few more now, looking at spatial transcriptomics in COVID-infected lung. Both of these previous papers published, mid-2020 and late 2020 used the DSP platform. But what was really interesting was when we dug in and had a look at their analysis methods, they were doing t-tests on the data. And obviously, being a bioinformatician, I wanted to sort of dig apart that decision and see whether or not there was a more sort of subtle way that we could look at analysing the data. So this is our experiment here. Arutha already introduced this very briefly to you. So we have samples from 3 different groups, 10 patients in each group, and these are samples that were collected at autopsy from patients who passed away in an intensive care ward in the Brazilian hospital. So these patients either had COVID-19, H1N1, which was a pandemic influenza, or had passed away from a non-viral medical condition. So these were our non-infected control groups. And we had 10 cores from the patients across these 3 different groups. 2 patients had particularly high viral loads. So these 2 patients here, when you have a look at the RNAscope for these patients, you can see these red patches here that are lighting up are showing high evidence of RNA from the COVID-19 panel. So we knew that these 2 patients in particular had a very high viral load when they passed away. And as Arutha mentioned, you can use the RNAscope data to guide your selection of regions of interest to sample both regions with high and lower viral load, but also look at individual tissue structures in the data. So what you can see here, on the right in this panel is that we have been able to select some quite specific tissue structures from these 2 cores and from others I'll show you in a moment. But you can see that these regions here have been selected to actually highlight particular epithelial structures, for example, or regions that showed interesting pattern of pathophysiology. So this guided region of interest selection really gives you a lot of power to start dissecting the heterogeneity within the tissue. Now, this was the sampling strategy across all of the 3 groups that we had. As you can see, there was obvious, much more heavy sampling over COVID-19 cause because of the interest in exploring the biology of these infected samples. And we had much lighter sampling over the pandemic H1N1 and the uninfected controls. Now, we learned quite a lot through this process, and we are using refined sampling strategies now in our further developments of this work. But nonetheless, you can also see that within cores from a patient, there are multiple samples that can be collected across different heterogeneous regions. So this is the data set that was collected. We were looking at the Cancer Transcriptomic Atlas with the 1800 genes, as it was described earlier, with an additional 26 added in COVID-specific genes. We had 2 negative controls, as well as a number of internal reference genes that we could look at with this. We had 60 regions of interest across 3 slides, each with 10 cores from either the COVID, H1N1 or uninfected controls. And we had 47 regions of interest from the COVID patients, 8 from H1N1 and 5 from our controls. So what did the data look like when we actually started to explore this? So we took the Q3 normalized data from the NanoString platform. Now the Q3 normalization uses top 25% of expressing genes. However, when you look at both a box plot of these data and more significantly an IRLE plot of these data, you can see that that Q3 strategy actually left considerable patterns of variability across this data set. So in particular, we were concerned about the fact that our normal patients, our controls here, all had substantially higher patterns of expression on an IRLE plot. And I'll come back to this later when I reflect on the choices that bioinformatics can actually have and the impact of that on your data. Now, if we look at TMM normalized data, on the other hand, we actually see that the IRLE plot indicates that we have actually removed most of that unwanted variation in the data. Here, the colors correspond to the different patients in our cohort, and you can see that the fairly heterogeneous impacts that we had here have now largely been removed by TMM normalization of the data. Now, if we look at exploring the actual data set itself to understand the patterns of variability that we see, we can see that our non-infected controls tend to cluster along PC3 here. So these are the square boxes that you can see here. These are the non-infected patients. Our COVID patients are in blue and our H1N1 patients are in purple. And you can see that on the third principal component of this PCA, we actually do start to get a segregation between normal and H1N1 and COVID, although there's some other interesting structures in the data that we wanted to try and analyze. There was also this really interesting cluster here that separated out on PC1 that we were quite concerned about trying to identify what was going on with that. Initially, I thought that might have been some sort of patient clustering, but it clearly wasn't. So if you look at the patients, we can start to see patient effects in the data. So these are repeated samples that have been taken from a single core of a patient, and you do tend to see patient clustering, in particular in this representation where we look at PC3 versus PC4. And these 2 cores here, LN1 and LN3, were actually our high viral load patients that were quite densely sampled for this experiment. But something that was really interesting was we didn't get to explain this particular cluster until we actually looked at the tissue structure. So when we got the pathology reports back that actually described the dominant tissue type of each of the regions that were sampled, we saw that this cluster that we noted here in the data actually corresponded to samples from the bronchiolar epithelium. And these actually represented one of the most dominant forms of variability that we saw in the data. If you look at all of these different visualizations of the variability, you can see this bronchiolar epithelial tissue has a very distinctly different pattern of expression to the rest of the samples that we had, irrespective of whether the patient was COVID, H1N1 or control. If we look at the viral load from the RNAscope, we can see that all of our high viral load patients corresponded to these 2 cores here. So that's LN1 and LN3, we've circled them here. They do tend to separate. But what's also interesting is if we actually have a look, unfortunately, all of our bronchiolar epithelial tissues came from non-viral high samples. So we didn't actually have any high viral load in these particular tissue structures, even though some of them did actually correspond to our high viral load patients. So after doing this sort of deep exploration of the data, it gave us a real handle on where we were seeing variability across the experiment. It was clear that there was variability between the COVID-19, H1N1 and our control samples, but it was also clear that the dominant tissue type was one of the main sources of variability in the experiment, particularly from these bronchiolar epithelial structures that we saw quite clearly separating, clustering out in the data. We also saw some separations based on high versus low viral load, and a little bit on the actual RNAscope score, although we didn't actually dig into that in any further detail. And so the considerations that we had to deal with when analysing these data were that we needed to account for the fact that we had repeated samples from a particular patient. You can see here the repeated sampling from individual patients is something that we need to consider, and that it was of differential depth. So we had 9 samples, for example, from patient LN1, whereas we only had 3 from LN2. We had to be able to make comparisons between and within cores because of the fact that we had different tissue structures within a core, and we needed to be able to think about how to deal with the similarities that we see within a patient when looking at differences between patients. And so we used the Lima pipeline to do this analysis of the data. So we did a number of different comparisons, and just to reiterate, in modelling the structure of the experiment, the things that we needed to take into account were disease type, patient core, dominant tissue type, viral load, and RNAscope. So these were actually a large number of variables for us to consider when we were structuring the analysis. So we looked at 2 main contrasts: COVID versus our control non-infected tissues, and COVID versus pandemic influenza. We used cores and dominant tissue types as covariates, and then we used the VUM-LINA pipeline to fit linear models to the data and do the differential expression analysis. There's a function in Lima called duplicate correlations that we use to take into account the correlations between gene expression within each core. And then we use the TREAT criteria as an additional criteria on the p-values to emphasize things with large fold changes in our results. So what do the results look like when we go through this? So of the 1,800 genes which had been selected, let's remember, for immune and cancer-related phenotypes, and in order to maximize the chance of differences, we saw a fairly substantial pattern of differential expression. There was not as much that went down, but we did see significant upregulation of a number of genes that are associated with immune-related responses, antigen presentation, fibrosis, for example, and the complement cascade. Now, many of these, particularly fibrosis and the complement cascade, are known to be part of the pathophysiology of severe COVID infection. So this was nice to see that those results were coming through as expected. Now, just to note, because we used the Lima pipeline, we were able to run differential gene expression analysis as part of that pipeline using FRI. And this is a summary of the pathways and gene sets that we saw in the top of our perturbed gene set list, just going down through, showing the difference between upregulation versus downregulation of genes in these pathways. And again, just indicating we saw quite a strong signal for blood coagulation, hypoxia responses, and angiogenesis in the data. Looking at the difference between the COVID patients and the influenza patients, however, we saw much less in the way of differentially expressed genes. There were very subtle transcriptional differences that we saw here. And initially we were a little bit concerned about this. However, talking to Christy, she reassured us that this is actually something that you'd expect. It accords with our expectations that the viral infections would have similar impact on the lung. The genes that we're highlighting here were selected by Christy. But what is actually notable is that there were a number of interferon-related genes that were upregulated in COVID-19 compared to H1N1 influenza, and particular IFI27 here which was upregulated with respect to our control tissues, was again strongly upregulated with respect to influenza. Now, it's possible that whole transcriptome coverage with the new whole transcriptome panel would show us a lot more here in terms of the sort of more subtle differences between COVID and H1N1. So I'm very excited to start working with that larger scale data set when those panels come out. Now, I'm just going to come back to, something that I observed at the start, which was that a number of the papers that are being published using the spatial transcriptomics platform have been using t-tests to model the data. And so we actually did a comparison between using the Lima VUM pipeline and using t-tests to analyze this data set, mostly just to satisfy my curiosity about how this would actually impact on the analysis. And what you can see is a number of the genes that were strongly detected in our Lima-VUM analysis were also detected using the t-test, but you can see a substantially different pattern of gene expression here. And particularly when you look at the COVID versus influenza, you can see that there was lots of significantly differentially expressed genes, all these triangular ones here, that were picked up in that comparison by a t-test that we simply don't see when we use Lima to analyze the data. And just coming back to the COVID-19 versus control, you might remember when I first showed you these data that I pointed to this upward shift in the relative log expression in our normal samples. Now, based on that, I would expect that something like a simple t-test would actually predict that things in the COVID samples were down with respect to those controls. And that's exactly what you see if you use a t-test and you don't take into account and correct for this sort of structure. So the t-test, or variations on it that are typically used in a lot of the papers, really fail to capture a lot of the complexity and the subtlety that we need to take into account when we are doing spatial transcriptomics analysis. So there's a number of arguments for using Lima. It's a very flexible analysis pipeline. It borrows information from genes across the entire experiment and can accommodate more complex experimental designs than what you get if you simply put your samples into two groups and do a t-test, which makes all kinds of assumptions about normality and independence of your samples and all of that sort of thing. And the low number of differentially expressed genes that we see in the COVID versus H1N1 is again expected. So, this is a fairly subtle difference between these two viral infections. And so we think this is a better reflection of what's likely to be going on. So in conclusion, the spatial transcriptomics profiling that we've done here, combined with linear models to look at differential expression, is a really powerful tool for identifying these novel gene signatures in tissues. The spatially resolved transcriptional landscape of infected and uninfected patients clearly becomes apparent when you look at the individual tissue structures that you're working with in this data. And we were really excited to identify IFI27 as a potential biomarker for severe COVID infection. It also distinguishes COVID from influenza. And as Arutha alluded to, we've recently validated this marker in an independent data set and shown it to be highly predictive of disease severity very early in infection. So this represents a potential biomarker that can be used to triage patients early in presentation and determine whether they're likely to go on and develop severe COVID-19. The tissue heterogeneity itself is vital to understanding the data because the difference in the transcriptomic profile of those tissue structures is one of the key signals that we see. And so taking that into account and correcting for it in the analysis was really important as a way of trying to identify the patterns that we were seeing between infected and non-infected lungs. And finally, representing all of the relevant factors in a complex experimental design like a spatial transcriptomics experiment really requires careful thought. As I said, t-tests and similar make some inappropriate assumptions about the structure of your data and don't account for that complexity in the experimental design. We need to think about correlated signal from within patient sampling and factor that into the analysis. Normalization remains a question that we still need to address, and it's something that my group is doing a lot more work on with the data that we're getting from both Rachel and from Arutha. And I can recommend Lima as a very flexible method for exploring differential expression because it can account for many of these factors and actually model them in the context of the analysis. RLE and TMM, followed by PCA to identify variation and dimensions of variability in the data, and then followed by Lima and VUM is the pipeline that we ended up settling on for this analysis. It certainly gave us the ability to identify, biomarkers that we've then been able to replicate in independent data. And I just want to sort of emphasize how exciting it is to be able to work with such new technology. There's always the opportunity for new and novel computational analyses from data sets like this. So it's been an absolute pleasure working with Arutha and the rest of the COVID team on this experiment. Just to wrap up, this is my marvelous team. So the work I've shown you today was led by Chinwe, who's a senior postdoc in my group. He did a lovely analysis of all of this, along with Damesh Buva, another one of my postdocs who was over here, along with input from Surur, who formerly a biostatistician in my group, now a PhD student. And the rest of my team, of course, are fabulous and constantly provide input and suggestions. The COVID team at the University of Queensland, I mentioned right at the very start, at the University of Technology, Queensland University of Technology. It's been led by Arutha, and his team, James Monkman, was really invaluable in us understanding and developing a good understanding of our data, and all of our colleagues over in Brazil. And of course, the spatial work I didn't talk about today has been done in collaboration with Rachel and Atsvind and David Ritchie's group, along with Ashley Ng, and we've really enjoyed working on those data as well. So it's been a learning curve for us, and I think we're getting some really great progress in understanding how to analyze these data. So I'll leave it at that. Thank you very much for your attention.
Thanks, Melissa. Beautiful work. Sounds like a classic case of speak to your bioinformatician before you design your experiment.
Absolutely. I would always say that, wouldn't I?
I've got one question in the chat from Anna Trigos. Did you notice any difference in the degree of intrasample heterogeneity between the infected and non-infected samples?
Unfortunately, the different level of sampling that we had between the infected and non-infected samples meant that it was very difficult to do that sort of analysis. So really the greatest depth of sampling that we had was for our COVID samples with a high viral load. Now we've done subsequent analyses after that where we're getting deeper sampling across the controls, and also doing additional sampling within our highly infected cores. And so that's giving us a slightly better handle on it, but we haven't finished crunching those data yet.
Okay, and I might follow up the LimaVum workflow, despite having an awesome name. How easy is that to implement as a lowly biologist, or is that something you have to work through with a bioinformatician?
Look, I would recommend working on that with a bioinformatics collaborator for a couple of reasons. It's not that it's difficult, and there are very good workflows that are available online that take you through how to use Lima in the analysis of gene expression data. But I think the thing that analysing these data really emphasised for me was the importance of actually developing a deep understanding of the data right up front and really digging into, how to model that complex experimental data correctly. We didn't have any idea at the start that the bronchiolar epithelial structures were going to look so different to the rest of the structures in the data. And, when you're getting very new data and you've never seen anything like this, it's hard to know what it is that you need to take into account. I naively thought that we would see that patient effects would actually be the strongest effects that we would see, but instead it was actually tissue heterogeneity and tissue structure. So this could just be because I know nothing about the lung, but it's also, it was an object lesson to me that you shouldn't make assumptions about what you might expect to see in your data. So I think because of the importance of actually getting that right and making the right decisions about how you capture the various different factors and dimensions of correlation in the data, it would be a very good idea to work with a bioinformatics collaborator on the analysis. But a bioinformatics collaborator wouldn't have too much trouble in replicating what we've actually done here. And I should say, we've got this work up on the medRxiv at the moment, so anyone who's interested can have a read through and see exactly how we've approached that bioinformatics workflow.
That answers the next question I was going to ask. All right, I think for the sake of time, we might move on. Thank you again, Melissa.
Thank you. Let me stop sharing.
Yeah, and we'll move on to general questions and answers in a second. I just want to very quickly just take you guys through the grant scheme that we're putting in place to celebrate the launch of the DSP. So within this, we have to thank NanoString and BioStrategy for their support in helping to fund these grants. So there are a few terms and conditions, and there's a link on the webpage at the bottom there, which takes you to the full terms and conditions. But these are open to researchers that are based at Victorian research institutes only. In order to apply for the grant, you need to submit a 200-word abstract. And the application period is open from today for a month. So we'll close on the 17th of June. I just realized I'm living last year, so it's 2021, not 2020. And the results will be announced on the 24th of June. So there are two sets of grants that are available. There are grants for the proteomics side, in which we will select 4 winners for multiplex protein profiling analysis, an 80-plex from the immuno-oncology protein panel, and that's valued at about 7,000 Australian dollars per winner. Each winner will be provided with reagents for their chosen topic for up to 3 samples, either FFPE or fresh frozen, with 12 regions of interest per sample. And these samples will be processed on the DSP system here at the Peter MacCallum Cancer Centre. For those that aren't successful on the protein side of things, we also have access to the whole transcriptomics. So one winner will be selected for a whole transcriptome analysis valued at greater than 15,000 Australian dollars for up to 2 samples, again with 12 regions of interest per sample. And those samples will be processed through the Technology Access Program via the headquarters in Seattle. All costs, including consumables, sample shipment, sequencing costs, and advanced data analysis are included within that. And the winners will receive a final data report by the end of September 2021. And also for those of you, if you don't get either of those, NanoString and BioStrategy are also going to support purchase of consumables for projects and basically matching funding. So 50% discount essentially for unlimited sample numbers, FFPE and fresh frozen samples. The discount is valid until August the 10th, and samples must be sent out no longer than October the 10th. Okay, so thank you again for NanoString and BioStrategy for their support of those grants. And I believe you will get an email after this event with a link that will allow you to fill in your submissions for those grants. That's everything from me. So let's open the floor to questions for anyone.
Hi, Paul. Thank you very much for sharing on the grant details. And again, NanoString would like to thank BioStrategy and Peter Mac Cancer Centre and also Royal Melbourne Hospitals for this event. And I had also posted a link in the chat for all the audience. If you are, as Paul mentioned, there will be an emails including the link and more details on the grant after the event. So please keep an eye out for that. And now I will open the floor for all the audience. If you have any questions, the room will be kept open for another 5 minutes for any questions that regarding the grant or regarding GeoMx DSP platform at the Peter Mac Cancer Centre Collab.
Vince, you have your hand up. You want to say something? You're unmuted, Vince. You should be able to speak.
Hi, Paul. Can you hear me?
Yep.
Okay. Yeah, I just wanted to say that if during the whole process anyone's got questions about the project or whatever it is, obviously, contact yourself. We at BioStrategy as well, we're here to help in terms of discussing project setup and things like that. So just reach out to either yourself or myself, and we'll get the ball rolling on that.
Melissa, did you want to talk over that question rather than just put it in the chat?
Yeah, look, I'm not sure if Wataya is actually still on the line.
Might be worth just for those that still
I think she is. Oh, they are. So yeah. So my thoughts on that, and I'm not sure if that was directed to me or to someone else. I think it really depends on the context of the question. So if you've got any exploratory data or exploratory imaging data on the sections that you're interested in looking at, that would help to start with. I think the thing that I would emphasize is trying to do even sampling as much as possible. If you sample certain structures from one patient, try and sample them from multiple patients. I think it's a good idea to sample heterogeneously within a patient as well. So it's really something though that it's impossible to provide an answer for without you know, knowing what the context of the question actually is. So I think having a chat with a bioinformatician or a biostatistician, someone who can understand what the likely impact might be, and also having your own understanding of where you think the variability and the heterogeneity will be in your data is pretty critical. There's a lot of data sets now that look at things like single cell analysis of particular tissues, either in mouse or human. So that could provide some insight perhaps into heterogeneity, even though it doesn't actually capture the spatial component that you're going to have to deal with these data.
So I see there are more questions coming, and again, we want to thank all the speakers and for supporting this event and sharing their data with us. And for the NanoString tech support team, we will stay on to answering more questions for the audience. And we want to thank all the speakers and also Peter Mac and Royal Melbourne Hospital for this event.